Hi, this is Miss Litton, and this is 7th period review. Say hi. Hi. Of our Unit 4. And for us, this is chapters 13 through 16. Chapter 13 is about control of gene expression. Yes? Yeah. Chapter 14 was about the history of evolution. No, was biotech. Biotech. And then chapter 15 was the history of evolution, and then chapter 16 was population genetics. Yeah, we hit them all? So I thought because we just finished chapter 16 and we learned some new content there that I would start my review with chapter 13 or 16 and then I'll jump back to 13. Does that make sense? Okay, so for chapter 16, let's start off with a question here. For number. Why is my phone talking to me? It's singing. Wait, really? Yeah, I know. Guess all right, red velvet cupcake. Oh. Okay, most of you got this. When there is a crisis, okay, then that's going to be bottleneck. Okay, some sort of crisis has to happen. When a small portion is the only one, not the entire population, but a smaller population gets separated from the larger population and they go off on their own way, that's called what? Founder effect. Okay, what else did people put? Let's see. A natural selection, no. No. Okay. You all right with this one now? Went over your wrongs? Okay. Good job, birthday cake. Hello, welcome. For number. Paper, pen, calculator. Creme brulee, ribs. Most favorable dessert. Yeah, genetic drift is always due to chance. What we just had a question on was genetic drift. What are the two categories of genetic drift? Founders and good. That's one way you can change allele frequencies. Please tell me other ways you can change allele frequencies. Let's remember our hand, shall we? What was the pinky for? And that means what? Small populations are more likely to experience genetic drift that we were just talking about. Right? Small populations undergo more genetic drift. What's the next one? Non-random mating. When you're purposely choosing, this is who I want to mate with. Okay, and what was the middle one? Mutations cause changes in allele frequencies. Movement, gene flow. Either immigration or emigration, and then most of all was natural selection that led to what? Adaptations. If you are in true Hardy-Weinberg equilibrium, that means that there is no change in allele frequencies, and what is not occurring? Evolution. Evolution is not occurring. But if the allele frequencies do change, if it disturbs the Hardy-Weinberg equilibrium and the allele frequencies do change, then what does occur? Evolution. Okay, what were the two types of evolution? Microevolution and macroevolution. Microevolution is change within the species, and macroevolution is change to an entirely new species. And that's chapter 17 that we're going to start tomorrow after our little testy quiz.
I'll watch it.
This should be fairly easy. Okay, I'm done. Yay, good job. All right, now, let's do a problem here. You ready? Yeah, let me get them started and I'll get, which chapter do I need to do first? 8? Okay, let me get them started on this. Okay, so here is a Hardy-Weinberg problem. I'm going to pause for a little bit. I'm going to tell you, you might have somebody next to you who wants to boss you and tell you what you should be doing and show you what they are doing because that's how they gain confidence. Tell them, stop. Work on your own. When you're done, give me a double tap on my desk and we will share. Okay? Or if you come to a halt, you can't figure out anything more, then double tap and be ready. But don't impose don't take away their ability to learn. Okay? I'll pause just for a minute. All right, so let's talk through how would we solve this. I didn't clickify this because there are multiple answers for this. So I'm gonna walk you through and then I'll have you fill in. So the first thing it says the allele for normal length wings is dominant over the allele for vestigial wings. If I were working this out on an AP exam, what's one of the first things I would write down? I'd write the genotypes and what they meant. So I would probably, because it's talking about wings, what do you think I would put for a letter? Yeah, so I would say big W anything is normal. And then what I put? Little w is what? Vestigial. That's the first thing I'd write. So in my Hardy-Weinberg problems, what letter is W actually going to be? P. And what's little w going to be? Q. Okay? So in a population of 1,000 individuals, 360 show the recessive phenotype. Okay, we have two equations that we deal with, right? What are our two equations?
P plus Q equals 1. P squared plus 2PQ plus Q squared equals...
Yep. Which one of those is talking about geno and phenotypes? The first one or the second one? What? That's perfect. Which one is talking about geno and phenotypes? First or the second? If I have a phenotype, what dictates my phenotype? Genotype. How many letters do I have in a genotype? 2. So which one on here shows 2 letters? This right here is representing homo dom. This right here, P squareds, would be like big W, big W. This right here is representing the heteros. This would be big W, little w. And this right here is representing the homo recessive, which would be little w, little w. That would be the first thing you want to understand. Okay? Now, if all of this population of fruit flies, if the whole population of fruit flies took their 2 alleles, because each one of them, it's diploid, each one of them has 2 alleles, right? And if they took both their alleles and threw them in the gene pool, and we counted up how many big W alleles and how many little w alleles, that would be the allele frequency, the P plus Q equals 1. Right? The percentage of big W's plus the percentage of little w's in our gene pool equal 1. That's what the P plus Q equals 1 is. I could talk about the allele frequency in the population and you could tell me what it is, but let's calculate it out. This says, in a population of 1,000 individuals, 360 show the recessive phenotype. That means 360 out of 1,000 have vestigial wings. which means that's your Q squared number. 360 divided by 1,000 is equal to what? Yeah, so Q squared equals 0.36. Do you understand where I got it there? So if I wanted to solve for Q equals what? 0.6. It's the first thing I would do. Oh, okay, Q equals 0.6. Now let's see what it asked me. How many individuals would you expect to be homozygous dominant and heterozygous for this trait? That means they want to know how many are P squareds and how many are 2PQs. So let's solve for the P squareds. Well, if Q is 0.6, I know, because P plus Q equals 1, I know if Q equals 0.6, then P must be what? 0.4. So homozygous dominants, P are these guys right here, right? These are my homozygous dominants, P squareds. So 0.4 times 0.4 is 0.16, which would be what percentage? 16%. What's 16% of 1,000? 160. So I'd expect 160 of them to be homozygous dominant. Then if I wanted to figure out how many of them are 2PQs, the heterozygotes, then all I have to do is say 2 times 0.6 times 0.4. 2PQ, right? 0.4 times 6 is 24, right? Yeah. Times 2 is 48. 48%, 480 out of 1,000. Tell the person next to you, how you feeling? Are you in the shadowlands or are you in the sunshine? Want to do another one? Yeah. Okay. Do this one. Again, I highly encourage you, do it by your onesie. Double tap when you're ready to compare with somebody. You have to take it by yourself tomorrow. I am not surprising you with a partner quiz, so don't even hope for it. It is not happening. Listen to me. It is not happening. Do you ever have me lie? No, I am not a pantalones and fuego. So make sure, do this by yourself. Did you guys see the beautiful sunrise I sent you this morning? Waiting for the comment of some student to go, Miss Litton, what are you doing using your phone while driving?
You were driving. I thought you were running.
I think I pulled over.
I thought you'd be running.
Running? No. I was on my way to work at that time.
Oh.
No. Running is for 3 in the morning, sweetheart, not 6:30. You're so confused. I have a slide that has all the answers to all 3 of these on it that you can capture, or I will PDF it, and it'll be connected to this YouTube video. Oh, I should have paused it. Can we go? Okay. Let me read it. Do you need a lot more time? Do I need to pause the video, or are we close? We close? Who wants more time? Okay, great. Okay, so in this one, the allele for unattached earlobes is dominant over the allele for attached earlobes. What do you think is the first thing we'd probably want to do? Yeah, I wanted to get some letters up here, so I'm going to go with E for earlobes. You have already done genetics. Do you see how you might have to do a genetics problem and then figure out Hardy-Weinberg? Do you see how that could work? Right in, floating back and forth. So big E, anything is unattached earlobes is dominant, is unatt. Little e is what? Attached earlobes. In a population of 500 individuals, 25% show the recessive phenotype. So that would be, this is the recessive phenotype. What else is that equal to? Q what? Squared, which is equal to 25%. So what is that? 0.25. So could I solve for Q? Yes, what is Q? Yeah, Q equals 0.5. What does that mean P equals? 0.5. Now let's read what the question says. How many individuals? It wants a number. How many individuals would you expect to be homozygous dominant? Homozygous dominant, what do we need? What are we solving for? P squared. Okay, so P squared, 0.5 times 0.5 is 25. So 25% of 500 is what? 125. So your answer would be 125 individuals. Do you see how somebody might accidentally on an AP exam bubble in 0.25 or 25%? So you have to read the question. How many individuals? 125 individuals. Because that's 0.25 times 500 people in this population. Okay? And how many would be heterozygous for this?
250.
Okay, because you did 2 times 0.5 times 0.5. Whoops. Which is 0.5, and half of 500 is 250. Ask your bio buddy, Shadowlands or Sunshine? You want one more?
Yes.
Hey, here's one more. I'm going to pause. Okay, we're going to be ready now. And before I even solve this problem, I want to remind you, highly suggested reading and thinking. It's on your test. Highly suggested, which you've been doing them as you go along, so that's not even stressful for you because you've been studying them all the time, so you're just going to reread through them to remind yourself of what you already know, because you are responsible students. Okay. All right, come back to me. In certain African countries, 4% of the newborn babies have sickle cell anemia, which is a what? Recessive trait. Okay, so 4% have, what part of our equation is that referring to? Q squared, which equals, if I'm writing it as a decimal, is 0.04. 4%, right? So if I want to find Q, what do I have to do? Square root, Q equals what? 0.2. That means P equals what? 0.8. Now, it asks me, out of a random population of 1,000 newborns, how many would you expect for each of the 3 possible genotypes? Well, we have healthy, we have carriers, and we have sick. Okay? These are your P squareds, 2 PQs, and your Q squares. Yes? Okay, so all I have to do then is go, okay, my P squared, that's 0.8 times 0.8 times, how many are in my population? 1,000. What is that number?
640.
What is it? 640? Okay, and then for my carriers, that's 2 times 0.8 times 0.2 times 1,000. What is that? 320? Okay, and for this one, it's just, we already know this, it's 4%, but what's left? 40? You all right with that? Okay, here is the answer for those 3 problems. Yes?
For all 3 of these questions, we were like, as our basic source of information, we were given the amount of recessive phenotype on the test. Will we only be given the recessive phenotype?
No. How else could I have restated this question? 96% of the population is healthy. That could have told me the same thing. On this one, what could I have said? 75% have the dominant trait. And then you would say, oh, 75% have the dominant, 25% have the recessive. Why do I always have you work off the recessive trait to solve for everything?
It's pure.
It's pure. It's the only one that's not got a mix. Because the dominant trait when you look at the dominant trait, you can get a dominant trait with P's and Q's. Okay? All right. Now, can I let that go? Can I move on? I want to remind you in chapter 13, do you remember this stuff?
Oh, yeah.
Okay, so we have operon, which is a prokaryotic cell type of a thing. An operon consists of a promoter, an operator, and the genes it controls. Remember? And there was positive and negative control. Negative control always had to do with a what? Repressor. And that repressor could either be inducible or it could be repressible. Inducible means the operon is what? Off. And I'm inducing it to start up. What do I induce it with? I could use lactose to bind to the repressor and pull it away. Okay, if it's repressible, it means that the operon is currently on, and I'm going to need to use a what to turn it off? Tryptophan, which acts as a co-repressor to bind with the repressor, activate it, and shut down the operon. Positive control is also used in prokaryotes, but primarily it's negative control. Positive control, you're working with a what? What's something that has to do with positive control? Transcription factors. Now, this is why in later chapters in genetic engineering, remember, or later, whatever, what's the next chapter? 13, 15, 14, thank you. Chapter 14, if you recall, we said if you want to get a eukaryotic gene expressed in a prokaryotic cell, we talked about how our eukaryotic DNA is full of what? Introns and things that get removed. So what do we have to do? We don't try to copy the DNA and insert our normal DNA in. What do we start with? RNA. What kind of RNA? mRNA. And what kind of DNA do we make from that mature RNA? cDNA. It's complex. Yeah, you're making DNA off of the messenger RNA. So that was one thing you had to do. And the second thing is we had to insert some sort of control regions. Controls that prokaryotic cells could understand. Prokaryotic cells understand operon. So if we're going to put a control region in, whatever vector we use, we've got to use something that will turn the genes on in a prokaryotic cell because we work differently. We work with transcription factors and positive control. And we have this, this, and this to turn on the genes. That's not how prokaryotic cells work. Okay, so they need an operon. They need something else brought in there as well. Okay, so we talked about when an operon is on, and then you can use something like tryptophan, which is what you were bringing up, to act as a co-repressor to shut it off and activate the repressor by binding to the operator. We also talked about inducible operons, where the repressor is already on the operator, and lactose. Since these genes right here, when they're transcribed and translated, they translate into a protein that digests lactose. You don't want to turn them on unless lactose is present. So when lactose is there, it will bind to the repressor, who lets go of the operator, and you will transcribe and translate those genes.
Yeah, you know, I remember
Yay! Yay! Okay, just tell me this because I want to review some other stuff. What's the part of the operon where the repressor binds? Operator, yeah.
Regulator.
Regulator gene codes for the repressor. Can I skip one? Yeah. Oh, I'm sorry. I thought I already answered. Name the part of the operon system that will bind to the repressor, making it inactive. Lactose is acting as a what?
Co-repressor.
Oh, it's an inducer. It's an inducer.
It's an inducer. Structural genes, good job. Okay, then we talked about levels of control, and we went into great detail on these things. So I don't have time to review all of those details with you, but I'm reminding you, you have chromatin structure. Is the DNA available or not? Remember heterochromatin, euchromatin, Barr bodies? Okay. Transcriptional control. Can RNA polymerase bind to the promoter? Is it negative control? Is it positive control? How is that happening? Post-transcriptional control. Put on a tail and out the introns before it leaves the nucleus. Translational control. Can it hook up with the small and the large ribosomal subunit? Post-translational control. The folding pattern imposed on the primary, secondary, tertiary structure. Yes? Okay. Then here. Five, 4, 3, 2, 1.
Yes.
Now, look at me. Look at me. We're here for a short time. Let's use it wisely. Okay? So let's logically think how we pick this answer. Some of you got it, but others struggled. So let's alleviate their fears, okay? So it's talking about getting the introns removed. What are you removing the introns from?
DNA.
Really? RNA. Okay, you're removing, cut out the introns before it leaves the nucleus. Who's leaving the nucleus? RNA, mRNA. So your mRNA is already made. You're not translating it yet. You're not on a ribosome. You're not building a protein. This is the processing that comes after transcription. Hence the name post-transcriptional control. Post-transcriptional control. Three, 2. Yay! Good. Chromatin structure. Good job. Remember we talked about epigenetic inheritance? And that has to do with chromatin structure, where the DNA is available for transcription or not. That would be right in there too. 3, 2, 1. If you are binding to a ribosome, what are you doing? What process? Translation. Okay? Remember your central dogma. DNA, RNA, right? Protein. What is this part called? Translation. What is this part called? And if the DNA is just making more DNA, what's that called? Replication. That's your central dogma. Okay? So if you're in the process of using your RNA to build a protein on a ribosome, that's translational control. If following this protein, you're folding it in some way, that would be what kind of control? Post-translational control. If you've made the RNA, but now you're snurping it, you're cutting parts out of it, you're capping, you're tailing it, it's got to leave the nucleus, that would be what? Post-transcriptional. Got it? Oh my gosh. Three, 2. Yay! Just answer me this. A protooncogene will always cause cancer? No, could. If it mutates to an oncogene. Okay, then we went into genetic engineering. What is something you use to cut DNA? Enzymes. They make what? I'll keep going if you keep staying. Okay, they create sticky ends. The beauty of that is I can cut organism A with restriction enzyme and cut organism B with the same restriction enzyme, and I will have the same what? Sticky ends. Then I can mix and match matchy sticky ends that are complementary and use what? Ligase to seal the phosphate sugar backbone. Why can I do this? I can do this because the genetic code is universal, right? And it doesn't matter what organism you're in, you can express that, right? An apple can express fish genes, right? If it has the right DNA. If it has the right DNA and you have amino acids, you can build it, right? Now, listen to me. You can't have a fish tail and a fish head be out of an apple. It's like swimming apples, bobbing for apples, okay? You are limited because there's several different genes working together to code for a tail and a fish. But something like a protein that makes the skin thicker, that protects the skin of the apple, you could put that gene in there, right? Because it's expressed in the same way. DNA is expressed as amino acids into proteins. Okay, so we looked at cutting and splicing DNA from different sources, and then we want to insert those into a host. Transgenic organisms, DNA from other organisms found within them. When we talked about transgenic bacteria, what else did we talk about? Transgenic plants and transgenic animals. And if you make multiple copies of that DNA or that animal, you have what? Starts with a C. Cloning. Good job. And from bacteria, you can isolate that product. Then this is the part I talked to you about expressing. Okay? You have to have a regulatory genes that the bacteria can understand. Like, you know, an operator, promoter, right? You've got to have that. You can't have any genetic junk in there, so the way to avoid that is to make cDNA from messenger RNA that we have. And then you need some vector to bring it into that cell. What are some good vectors? We talked about two. Plasmids and viruses. Good. Okay. Oh, we already talked about that. We talked about different skills. We talked about PCR. PCR is the polymerase chain reaction. Make multiple copies of DNA that you need that you might want to study. So if you're going to do that, what are some things I need to put in my thermocycler in order to do PCR? I need primers so I know where to start. Bases, my ingredients. I need some TAP, polymerase, DNA polymerase that can withstand those changes in temperatures that I get from Thermus aquaticus, right? Okay. All right, and that's PCR, multiple copies. What if I want to analyze the size of fragments? What will I use? I will use gel electrophoresis. That can give me a DNA fingerprint. The smaller the piece, the farther it travels from the well. And I can compare DNA fingerprints. You can't see exactly where it cuts, but when you see it out in the well, then you know this piece farther from the well is shorter and this piece is what? Longer, because it's closer to the well, so it moves more slowly. Okay, okay, okay. Okay. I don't know what to do. Okay. So if I want to introduce someone, I just need to be able to cut and tape. Why would I need DNA polymerase? To copy. I'm not talking about that, right? I'm just introducing a foreign gene. So I don't need any DNA polymerase. don't know what to do with your brain.
Can everybody sit this down?
Because it's nature.
5, 4, 3, 2. All right, good job. All right. Who had the smallest fragment of DNA? Suspect 1, 2, or 3? 2. You're assuming the well holes are here and the DNA is traveling. Now, it didn't tell you which ones, but what? No, because samples 1 and 3 didn't have it. Oh. Yes. Five, 4, 3, 2.
Are you serious?
Yeah, just A. Just A. What's the word you look out for in B?
Virus is butt.
Yeah, but, sure. Look out for a big butt. You know you have 2 vectors. What are your 2 vectors? Plasmids and viruses. Do they carry only the foreign gene? No, they could carry their own genes. Only gene therapy uses vectors? No. Genetic engineers are not doing gene therapy. What's the answer? A. You don't need this one. Ex vivo, in vivo. Which one do you remove the cells, alter, and return? Yes. Okay. Which of these? B. Plants aren't vectors. Who's a vector? Plasmids and viruses. I don't know how many times I say that. Okay, that brings us to chapter 15. So you want to think about change over time. Okay? You want to think about things like biogeographical evidence. We talked about several other pieces of information that Darwin used. Remember those tortoises? Okay? This brought us to natural selection. What's key in natural selection? Key. You gotta have this. Variation. Yeah, you gotta have variations for natural selection to work. Look at me. If they're all exactly the same, is natural selection going to work on them? No. And selection can only work on things that are expressed in the phenotype. Right? Because you can hide a gene if you are a what? Yeah, if it's recessive in the heterozygote, you can hide it and you can persist. Even if it's a bad gene, it can persist in the heterozygote. All right? And then we looked at evidence for evolution. Fossils, biogeographical, anatomical, biochemical development. So you want to look through those because that's recent, so I expect you to know that a little bit better. Okay? Here, let's just answer this. The flipper of a dolphin and the fin of a tuna. What do they have the same anatomy? No, so they are not homologous. Do they have the same function? Yes, they are what? Analogous. Analogous structures only. Like an analogy. The wing of a bat and the arm of a human. Do they have the same anatomy? Yes. So they are what? Homologous. Are they analogous? No, we don't do the same thing with bats. So they are, what one are they? A, homologous structures. What is a vestigial structure? What is a vestigial structure? That's what I'm asking you. Yeah, something that you used in your ancestral past, which doesn't contribute to your fitness currently. Why do we study vestigial structures?
Find a common ancestor.
Common ancestors. Right, it's evidence of evolution. Good. Natural selection occurs when the better adapted members of a population reproduce. Yes, I've already asked you that. Yes, true.
False.
Oh, and I guess that was it. We did them all. Yay! Super smart. Review. If you're really tired, have a piece of toast with butter on it. Go to sleep.